Okay, this is still in Chapter 5 for General Biology, still under Section 4 within Chapter 5, and we've gone over the process of diffusion. Now what we're looking at is osmosis, and osmosis is a form of diffusion. It is a form of diffusion where it's the diffusion of water across a selectively permeable membrane. And since it is diffusion, that means that the cell does not have to do any work to cause the movement of water to happen, that it happens on its own. So it is a form of passive transport. Remember, we have passive transport, which means that the cell does not utilize any of its energy to cause this work to happen. And we have active transport, which means the cell is utilizing its energy to cause the movement to occur. This one is passive. Now then, along with osmosis, we have the term tonicity. Tonicity refers to the amount of solute, or solutes that dissolve stuff like salt or sugar, the amount of solute that we have in the solution compared to the amount of solute inside the cell. And so we've got three possibilities. We've got greater than, less than, or equal to. That is, the amount of salt or sugar, the amount of solute out there in the solution can be greater than what's inside the cell, could be less than what's inside the cell, or it could be equal to what's inside the cell. Okay, so that's our three possibilities. Now then, I'm going to look at a diagram here, and let's go right here. Well, maybe. There we go. Okay, so yeah, here we go. Okay, so this is a beaker, and we've got a solution in there, and there is a hunk of tissue or a giant cell. Now in this cell, we're going to say that we've got 0.9% sodium chloride, 99.1% water. Okay, now our solution out here could have the same thing. We could have 0.9% table salt or sodium chloride, 99.1% water. Okay, now remember, for diffusion, we have to have a gradient. We do not have a gradient here. We've got the same amount of salt on both the inside and the outside of the cell. We've got the same amount of water. So we are not going to have any movement of the water. Now the salt or sugar does not freely go across that membrane. It's the water that will freely go across, and the water it continues to move, but it is an even exchange going into and out of the cell. So we've got a name for this solution. The term iso means same as, so this is referred to as an isotonic solution. Okay? And this solution is ideal for animal cells. Okay, so ideal for animal cells, because under these conditions, an animal cell is not going to gain water or lose water. It will maintain its normal size and normal shape. So this is what would have ideal for the animal cell. Okay. Now let's go back over here. And here is a beaker. Have a solution in there. We've got a hunk of tissue or a giant cell, 0.9% sodium chloride, 99.1% water. Okay, now I keep making the cell 0.9% sodium chloride, 99.1% water. One of the things to realize, the cytoplasm of that cell is not just table salt and water. It's made up of all sorts of proteins and all kinds of carbohydrates in there, all sorts of different minerals in that cytoplasm, but all of those materials taken together have a pull for water or an osmotic potential equal to a solution that is 0.9% sodium chloride, 99.1% water. So that's why we're using that as a fluid in the cell. Now, our first solution was isotonic. Have the same thing in the solution as in the cell. Well, our next possibility is less than. We could have only 0.1% sodium chloride out here in the solution, which leaves room for 99.9% water. Okay, so this solution is termed as hypotonic. Hypo means below, and that's referring to the amount of solute in the solution compared to what's inside the cell. Now, we do have a gradient this time. Water is going to move from where the water is in a high concentration to where it's in a low concentration, or another way to look at it, water will move to where we have more solute. Either way, it means that water moves into the cell. Water takes up space. This is going to cause the cell to enlarge. In animal cells, this could possibly be a problem. So in an animal cell, we could get what's called cytolysis. This is where the cell expands until the cell actually just bursts open. And then, of course, it dies. If we're talking about red blood cells, we have a special term for that. It's called hemolysis. Now, for plants, this is a pretty good situation because for plants, they have that reinforcing wall around the outside, and so this is the ideal situation for plants. So, it doesn't work so well for animals, but it does work quite well for plants. Okay, let's go to another one here. So, we've looked at equal to, less than, isotonic and hypotonic. Let's look at greater than. So, here's our, if I can get it to draw. Okay, here we go. So here's our beaker. We've got some solution in there. We have a giant cell or hunk of tissue that is 0.1% of 1%, 0.9% sodium chloride. If I said 0.1% sodium chloride on either of those last two, that was a mistake. It was supposed to be 0.9% sodium chloride, 99.1% water. Okay. Now out here, this time our solution is greater than. So let's say 5% sodium chloride, which leaves room for 95% water. Okay. The water is going to move from where the water is in higher concentration to where it's in lower concentration. So the water is going to move out of the cell. Water takes up space. This means as the cell loses water, it will shrivel up. If it shrivels up enough, if it loses enough water, it will die. So this would apply whether it's plant or animal. And so we'd say the cell is undergoing a process called plasmolysis, or that the cell is becoming plasmolyzed. Okay, so we would refer to this solution as hypertonic. Hyper means above, and so the amount of solute out there in the solution is above what we have in the cell. Hypertonic solution. Okay. Okay, so let's go over here. Now, here's a diagram in your textbook, and you can see in this first one we have an isotonic solution. We've got equal number of the little blue dots represent the water, and same thing over here. The green dots represent some kind of salt or sugar. So both the water and the solute are in equal concentrations on either side of the membrane. There is no net movement of water. and the cell maintains its normal size, normal shape, and you can see these red blood cells right here with their normal shape kind of being dished out in the center. Okay, this next one is a hypertonic solution, and hyper means above. The amount of salt or sugar, the amount of solute represented by the little green balls, is greater on the outside of the cell in the solution than what we have here in the cytoplasm. So water moves out of the cell. The cell shrivels up due to the lack of water. It undergoes plasmolysis, and we had actually, a term I didn't give you a minute ago, but since these are red blood cells, notice how as they shrivel up in a very controlled manner. The surface becomes crenate. So we have a term for that. They undergo crenation. So that's a term that would apply to red blood cells undergoing this plasmolysis. Okay, this last one is a hypotonic solution. Hypo means below. The amount of salt or sugar, the amount of solute out here in the solution is less than what we have inside the cell. So water moves into the cell, and the cell gains water, and you can see some of these red blood cells right here that look somewhat inflated, which they are. They're inflated with water. If that continues, they will go to the point that they burst, and that is referred to cytolysis, or more specifically for red blood cells, hemolysis. So plants will do quite well because they have the reinforcing cell wall. They'll do quite well in hypotonic solution. Animal cells will not. Animal cells need an isotonic solution. Okay. So let's go back over here. Try this. Okay. Here it's showing a plant that's being watered. So we're generating a hypotonic solution. Water can enter the cells right here, fill up the cells, fill up the vacuole, and produce what is called turgor pressure. Turgor pressure is the water pressure inside those cells. So we'd say that these cells are turgid, which is just fancy word for water filled, or that they have a turgor pressure. And then as we allow the soil to dry, well, it starts creating moving from a hypo to an iso to a hypertonic solution, which actually pulls water out of the cells, and the contents of the cells shrivels up, undergoes plasmolysis, and if that goes far enough, those cells die. Okay. Okay, so when we look at osmosis, human red blood cells and most other mammalian cells are isotonic with a solution that is 0.9% sodium chloride. So you go down to the pharmacy, you purchase eye drops, or you get a saline solution. This is what it is: 0.9% sodium chloride, 99.1% water. It is in balance with the mammalian cells. And again, that is not saying that the cytoplasm in mammalian cells is 0.9% table salt and 99.1% water. It's saying if you take all the contents of that cytoplasm, all the different minerals, all the different proteins and amino acids, carbohydrates and everything, that has a pull for water equal to a solution that is 0.9% sodium chloride, 99.1% water. Okay, and then this is what we just talked about a minute ago. Animal cells will function best in an isotonic environment. Plant cells function best in a hypotonic environment. Okay. Now then, water crosses membranes by two means. One is by sliding in between those phospholipid molecules, finding gaps in those phospholipids, sliding across there. The other, and which is probably the major means of moving water into and out of the cells, is by moving through these channel proteins called aquaporins.